how to manage and control gastrointestinal nematode parasite in small ruminants. One of the most important diseases of small ruminant industry, which we often overlook, I will say, is the gastrointestinal parasitism. So parasites, they have a very peculiar ability to live and to let live. They will do anything to survive, and they will not like to kill their host as well. So they will cause harms to an extent that the owner is unable to perceive. But if you see over a long period of time, these parasites cause a very high economic losses in terms of reduced working capacity, lowered fertilities, lot of culling of animals because of lower weight gain, lowered production, lower poor quality of wool, then lowered fleece weight, and of over and above, add to this the cost of treatment that is involved. There is practically no farmer who is not feeding anthelmintics to his animals. So that's how they cause immense economic losses to the pharma industry. So in today's lecture, I will be covering about only the gastrointestinal nematodes of small ruminants, and that too in Indian scenario. Now, if you see, there are at least 20, 30 species of gastrointestinal nematodes that affect animals. But the major ones I have listed based on the different agroclimatic zones that we actually studied. In hot and humid climate, Haemonchus contortus, followed by Punostomum, Trichostongolis, Esophagostomum, Puperia, and Ostatagia. These are the important parasites. These in hot and humid climates, they are actually the breeding houses for these gastrointestinal nematodes. If you go to hot and dry climates, you will again find Haemonchus there. You will find Trichostongolis there, and another important is Esophagostomum there. If you go to the cold climate, again Haemonchus will be there along with the other. You go to coastal areas, you will have Haemonchus there. In your area, all Haemonchus is the most important parasite. In variable regions, you go anywhere, you will find this parasite, Haemonchus contortus. Along with, in most of the cases, you will also find Esophagostomum. 1, 2, and 3. The third, I will say, is Trichostongolis. Haemonchus, Esophagostomum, and Trichostongolis. These are the three most important gastrointestinal nematodes that cause harm or cause a very important clinical diseases in animals. Life cycle, the diagram that I have shown will represent all gastrointestinal nematodes. They have a direct life cycle. They do not involve any intermediate host in their life cycle. So the life cycle is very simple, and that's why the parasites can easily propagate. The parasitic stages, that is the eggs, so these are passed out in the feces of the host or in the droppings of the host, like sheep droppings. So they will be passed out. These eggs, they are quite resistant. They can survive extreme conditions for few days, but not for very long. They require moisture, a temperature of around 27 plus minus 3, 4 degrees, plus presence of oxygen to develop a larvae within them. So as soon as these eggs are passed out in the feces, the development, the embryonic mass that is present in the egg will start dividing, and a first stage larvae will be produced within the egg. Now, this first stage larvae will then convert into a second stage larvae within the egg itself. And finally, a third stage larvae will be produced within the egg. This third stage larvae is produced, the egg will hatch, and the third stage larvae will be released. This third stage larvae will be available then on the pasture or on the soil. And the third stage larvae is actually negatively geotropic and positively phototropic. So in the early morning hours, when there is mild light, this larvae will crawl on the grass blades. Another important thing is that these larvae, they are also attracted towards water. So in the early mornings, you will have dew on the grasses. So all these larvae that were present in the soil, they will crawl slowly and slowly up and will enter into these water droplets, and they are then the source of infection for the definitive host. But if the temperature is high, if it goes beyond 35 degree or 30 degree, 33, 34 degrees centigrade, and there is dryness, these larvae will dry off. So once they climb on the pastures, they are then easily accessible for the animals that are grazing on that grassland. And once these larvae are ingested, they reach to the abomasum, where they develop into fourth stage larvae, and finally the fifth stage larvae from the which are also known as the young adults. The fifth stage larvae will then convert into the fully developed adult parasites. So this is a very simple life cycle. You do not require any intermediate host, nothing, only a little bit of ambient climatic conditions and some grasses on the ground that are sufficient for the development of the parasite and for its transmission to the next host. So the important is that the third stage larvae, they are the connecting link between the free living and the parasitic stages. The third stage larvae are actually free living, and they actually then infect the definitive host, and they get converted to fourth stage and fifth stage larvae, thereby producing parasitic disease in the animals. So this was in very brief about the life cycle. Now let us talk of the different important gastrointestinal nematodes. And the first important gastrointestinal nematode is the abomasal worm of sheep and goats, which we call as Haemonchus contortus, which is also known as the barber's pole worm. It is also known as the bankrupt worm. So you can see the importance of this where it can cause bankruptcy. So to the economic losses that it can cause can be up to that level that it can make a animal owner bankrupt. It infects mainly sheep and goats, and it parasitizes in the abomasum. Now, it is a very important or highly pathogenic gastrointestinal nematode because it is a voracious bloodsucker. Each Haemonchus contortus in a day, one adult parasite in a day can suck about 0.05 to 0.06 ml of blood. Imagine if there are only 1,000 worms in an animal, then how much, about 50 to 60 ml of blood is lost. And if about 10,000 animals, almost half a liter of blood is lost in a day. They can suck that much amount of blood, and this results in severe anemia in case of hemochromatosis. Acute infections, they generally occur when the sheep and goat, they ingest a large number of third stage larvae in a very short span of time. Let us say I'm saying that 10,000 eggs is the infective, 10,000 larvae, third stage larvae are the infective dose. Now, if a sheep ingest these 10,000 larvae within, let us say, in a day, in the morning hours when it preys at one time, then it will produce a severe disease. But the same sheep, if it ingests these 10,000 larvae over a period of about 7 to 10 days, then it will result into a chronic disease because a lot of immune response is also generated against these larvae and their stages when they molt in the body. And this helps in production of immune response also. But economic losses will not stop because of this. So there will be a lot of economic losses because of chronic infections as well. The clinical signs in general will include anemia because they are voracious bloodsuckers. There is excessive loss of blood. There will be hypoproteinemia in the animals, and this leads to bottle jaw condition. Along with this, there will be diarrhea. In the acute cases, particularly, if it is a chronic case, then diarrhea followed by constipation, then again diarrhea, this kind of syndrome will be seen. And the animals will have reduced appetite, and over a period of time, they will lose weight and production losses will occur. Now, if such animal dies and if you conduct the post-mortem, then you will find there is anemia. There will be paleness and whitening of different organs. And it is this anemia is quite apparent in the conjunctiva. You can see it in the live animal as well. Conjunctiva is normally pinkish to reddish in color, but because of severe anemia due to hemochromatosis, it will become pale or whitish in color. Then there is edema of subcutaneous tissue, hydrothorax, hydropericardium, all associated with hyperproteinemia. And because it is a bloodsucker, wherever the worm parasite attaches to the abomasal mark, you will find hemorrhages. Generally, focal areas of hemorrhages will be seen. If a animal which is clinically ill, animal will show clinical signs of the disease when it has about 10,000 to 12,000 worms in its body. And severity of the disease is actually the function of the number of the worms. How many worms are there in the body and the size of the animal. If it is a very small animal, then 10,000 larvae will be sufficient to, or 10,000 worm will produce a very acute clinical disease. But if the size of the animal is large, then maybe large number of worms, a little bit larger number of worms will be required. Now, that can be easily seen in the next line, that in lambs, 2,000 to 3,000 worms, they cause a heavy burden. And whereas in adults, it is up to 10,000. So the number of worms as well as the size of the animal matters. So a 2,000 or 3,000 worms can produce a clinical disease in lamb, but up to 10,000 or 12,000 adult worms will produce a clinical disease in the adult animals. The first photograph, this one, is a flock of sheep. You can see they are grazing on the ground and they are grazing very close to the ground. And they are debilitated animals. Shearing, yeah, all they have been sheared, but because of shearing, you can see the condition of these animals. They are all debilitated and they are grazing together in a flock. Now, if you pick one of these animals and you see they were showing the clinical sign of diarrhea as well. And if you see that there will be pasting of the feces in the posterior part as shown in the second photograph. You can see the feces, they are watery and they are pasted on the perianal region. So in a flock, you will find a large number of animals showing this kind of symptom when there is acute hemochromatosis, or even in chronic infections, you will find this diarrhea. In the early morning hours, you will see flocks of sheep going for grazing, and they will be passing out their droppings, the pellets, they will be on the ground. But in a flock which is suffering from hemochromatosis, a clinical hemochromatosis, instead of pellets, you will find a lot of direct feces that are present on the ground. And this diarrhea will not be seen in one or two animals. Almost 20, 30, 40% of animals will be showing this diarrhea in a flock because these animals are grazing together on the same pasture land, on the same ground, and they will be ingesting the larvae almost together. So a large number of animals will be showing this diarrhea. Then the third photograph that I of a goat, and you see the conjunctiva of this goat. It is totally whitish. It is totally anemic in nature. So this animal, and along with this, you can see there is a little bit bottle jaw, which is more prominent in the fourth photograph, this bottle jaw condition, as if a bottle of water has been inserted in the neck region. So these are typical clinical signs in case of hemochromatosis and in GI nematodosis in general that you will be seeing. Now, if you conduct a postmortem on this, one of these animals, you see the first photograph, the number of Haemonchus contortus that are present in the abomasum in a small area. See, maybe not less than 10, 20,000. There are bunches of the parasite and they have all sucked blood. That's why they are red in color. The intestine is full of blood and that's why it appears red. And you see, you think of a situation of this animal. This animal died because of hemochromatosis and because of severe anemia. And then we separated few of these parasites, not all. And we place them in a petri dish. This is from a very small part of abomasum. Then the third, these are the eggs of Haemonchus contortus that I will discuss later. Then the second parasite of importance is the nodular worm, which is known as Esophagostomum columbianum, the species is that infects the cecum and column of sheep and goats. And it is known as the nodular worm because it produces small nodules in the small intestine of sheep and goats. And that's why the disease is known as the nodular disease, or it is also known as the pimply gut. The nodules are looking like pimples on the gut. That's why the pimply gut. The clinical symptoms include persistent diarrhea with dark green feces. The feces are dark green in color and diarrhea is persistent and it contains a lot of mucus and blood. Other clinical signs: alternating diarrhea and constipation, emaciation, general weakness, atrophy of muscles occurs, and finally complete prostration, and death may occur in some cases very quickly, within 1 to 3 days even. If you conduct a postmortem on such animals, you will find large number of nodules in the intestine, but underline it that the adult worms are absent in such cases, because the larvae of the parasite is responsible for production of the nodules. The fourth stage larvae will be, if you open this one of the nodules, we will find the fourth stage larvae in the parasite, of the parasite in this node. And these nodules will only be found in those animals which have already been primed to this infection at least once. Means if a lamb gets the infection for the first time in its life against this parasite, then there will be no nodule formation in that case. All the larvae will get converted into adults and they will release eggs in the feces. But if the same animal gets reinfected with the Esophagostomus columbianum, then at that time, the nodules will be found by the fourth stage larvae, which actually penetrate into mucosa of the small intestine of this sheep and goats. And because of the immune response, the host is already primed against the parasite. So because of the immune response, the nodules are formed in only such kind of animals. And these nodules, if they rupture towards the peritoneum side, it can result in peritonitis, or it can also result into marked adhesions. So these are the photographs of Esophagostomus, the nodular worm. You see the first photograph, the pimply gut. You see the small nodules present on the outer wall of the small intestine. These are all looking like pimples. And this is why this whole gut, because the disease is known as the pimply gut. If you open the small intestine, you will find the adult worms in the host, and you can collect the small intestine. The third important parasite is Trichostongolis, which is again found in the abomasum of sheep and goats, and is responsible for anorexia, poor growth, and passing of soft feces. This may be accompanied by diarrhea and weight loss. The important thing is the pathology produced by these parasites. These parasites can produce ringworm-like lesions in the abomasum, or it may produce Morocco leather-like lesions in the intestine. Morocco leather or ringworm-like lesions. And because of these ringworm-like lesions produced by Trichostongolis species, the parasite, the feces in some of the species of Trichostongolis will be blackish in color, and that is why it is known as the black spore worm also, because of the blackish feces that are passed out in this case. So this photograph, the first photograph is of the ringworm-like lesions, and you can see these blackish worm-like, brackish ringworm-like lesions in the abomasal mucosa of a sheep. The second photograph shows these raised areas in the abomasum. These are not too many here, but if they are their number is very high, then it will give an appearance of a typical Morocco leather. The character of Morocco leather is that it has got raised areas on the surface. So here also in the mucosa, you can see these raised areas like small nodules. They are present in the mucosal wall of the abomasum. And if their number is large, then it will give an appearance of a typical Morocco leather. Another important thing is that these parasites are very small in size. And while examining the abomasum, you may miss them if you are not examining it very carefully. They are very small, very thin, and whitish in color. So there are all chances that they may be missed in the routine postmortem examination. However, the lesions produced by these parasites, they can be easily seen. Then another common gastrointestinal nematode that is found in Indian sheep and goat is the Trichuras, the whipworm. which is found in the ceca of sheep and goat again. Now, it is known as the whipworm because it appears like a whip. The anterior end is thick while the posterior and the tail portion is very long. The anterior end, it tunnels into the mucosa of the small intestine, and it works like a stylet to cut the tissue and also the blood vessels. So wherever this parasite will enter into the mucosa of the small intestine, you will find a pool of blood also there in some of the cases, or a lot of mucus will be there because of the strong immune response that is produced there. The clinical signs, because it will cut the blood vessels and all of those things, there will be anemia, there will be dehydration, weight loss, anorexia, and the mucosa will be edematous and have catarrhal inflammation. You can see these are the trichuras. So these trichuras, they are embedded into the mucosa of the parasite, though the whole trichuris is like this in the second photograph. One end is broader and the remaining part is like a hunter. It is just like a hunter. So it will enter into the mucosa and the remaining part will remain out. It will cut the mucosa as well as the blood vessels. If few numbers, it will produce edema and catarrhal inflammation. If very large numbers and lot of loss is there, then it will cause pools of blood in the intestine as well. And the eggs of this parasite are typical. They are, I will say, barrel shaped with plugs at both the ends, and they are brownish in color. Another parasite that is often missed during postmortem examination is Strongyloides. Again, a very small parasite. It is found in the small intestine. It is known as the threadworm, and it can cause erosions in the intestinal mucosa, and the gut contents will be watery. There will be diarrhea along with moderate anemia. And important thing is that this parasitic infection occurs by the penetration of the larvae into the skin of the host. So when the larvae penetrates into the skin, it results in the introduction of the causative agent of the foot rot. And the animals infected with Strongyloides papillosus do sometimes suffer from this condition, that is the foot rot. This is a egg of Strongyloides. The character of the egg is that when passed out in the feces, egg is having a larvae, well-developed larvae inside it. While in all other cases that I discussed before, the egg the larvae develops once the eggs come into the environment. But in case of Strongyloides, the eggs, they have the larvae, developed larvae inside it when they are passed out in the stools of the host. I told you the clinical signs, they are almost overlapping in all the cases. It becomes difficult to tell whether the animal is suffering from Haemonchus contortus or from esophagostomum or others. So you should have some knowledge on the pathogenic potential, which I discussed in the previous slides. And you should also know which parasites are common in your case. That will help in the diagnosis. That will give a clue what kind of infection it is there. And you should always be able to distinguish helminthiasis from helminthosis. Means you should differentiate the clinical disease, the animals showing clinical signs, and the animals which are merely acting as the reservoirs or the carriers of the infection, and they are not showing clinical signs. Typical lesions are important, so three criteria should be followed. One, the lesions, the presence, the clinical signs, the number of parasites that you can see, and the pathogenesis that they produce. If you have this knowledge, then only you can reach to some kind of diagnosis for gastrointestinal nematodosis. In general, clinical signs, it is difficult, but because none of the clinical signs is pathognomonic. I talked of bottle jaw condition. It is seen in case of fasciolosis. It is seen in case of amphistomosis. I am talking of parasites. You talk of diarrhea, it can be bacterial, it can be tuberculous, it can be seen in fasciolosis, it can be seen in amphistomosis. Weight loss, any of these along with malnutrition. Clinical signs are actually overlapping in so a laboratory diagnosis is important. And the best tool for laboratory diagnosis is the fecal examination. Collect the fecal sample. You can go for direct examination of feces and demonstrate the eggs. Or if it is a chronic infection, the number of eggs are very low, then you can go for concentration. You can go for flotation methods. Then second thing that you require, whether you should opt for treatment or not. You have seen the eggs. If it is a hardly 1 or 2 eggs are visible in the fecal sample that you were examining, in the slide that you were examining, then you may think, why to treat such animal? It will help in production of immunity. In such cases, you should also go for fecal egg counts. It is generally said that if the fecal egg counts are above 500 in sheep and goats, then you should go for treatment. And 500 egg counts means there is a slide known as McMaster slide. If you use that slide and if you find hardly 10 eggs in that slide, then it means the EPG, eggs per gram of feces, is around 500. So an EPG, eggs per gram of feces, are above 500, then you should definitely go for treatment of such animals. Next thing is also important. You should know about the fecundity of the parasite. And you should also know about the biotic potential of the parasite, how fast the parasite proliferates, how many eggs it produces. These things also matter. There may be a parasite which is producing, which can produce large number of eggs. And there are hardly 10-20 parasites in the intestine, and it is producing eggs. They are coming, EPG is coming above 500. Then do you require a treatment for such animals? That is what you have to decide. And that you decide what you want, whether you want to treat this animal or you want to prevent the spread of the disease because of this animal. For me, I will like both of these things to happen. I will not want that my animal should be sick, and I will not want that my animal does not, the spread does not occur because of one animal which is producing large number of eggs. So I will, in both these circumstances, I will go for the treatment of such animals. And that's why if the EPG is above 500, whether one parasite is present or a large number is present, on my personal thought, I think that we should go for a treatment. Then the eggs of the parasites, these gastrointestinal nematodes, they are almost alike. They are generally called as the Strongyloides. They are almost similar. So to know which species of or which genera of these GI nematodes is present, you can go for coproculture. You can culture the feces in the lab, isolate the larvae, the third stage larvae. They have certain characteristic features on which they can be differentiated, but this required a lot of training. All of us, we cannot do. We do it in our labs because we are already trained for this. See, I have shown few eggs. The second, third, fourth, fifth, Haemonchus, Esophagostomum, Bunostomum, Trichostongolis, all are almost similar. Almost similar. They are all known as the Strongyloides. The first one is clear. It is Strongyloides because when passed in the feces, it is having larvae. The last one is clear. It is Trichuras, barrel shaped with plugs at both the ends. But the four between, they are almost similar eggs with very minute, I will say, morphological differences. And that again needs a lot of expertise to differentiate these eggs. So you go for coproculture and you examine the larvae in all these cases. Larval identification itself is a difficult task. You have to identify the intestinal cells, the shape of the intestinal cells, the number of the intestinal cell, how the mouth cavity looks like, how the esophagus looks like, and how the tail of this parasite, the larvae, looks like. And a combination of all them will help in the identification of the larvae. So I'm not going into details. You can see it, you can keep it as a guide with you and later on use it. Now comes, once you have diagnosed the disease, the important thing is the management and control of the infection. The foremost thing is proper environmental management or good managemental practices that are required. So good control requires a good management and a little bit of common sense also. I told you the larvae of the parasite, the third stage larvae, they are the infective stages, and they are most active during early morning hours when there is very low sunlight and there is dew on the soil. So all the larvae will graze on the grasses. They will get collected into the dewdrops, and when a sheep or goat will graze on sheep will graze on them, then they will ingest these larvae. Very easy. So we should avoid the grazing of animals during these hours. So allow grazing after sunrise and before sunset so that the dewdrops are dry during the daytime and the larvae will retreat back into the soil, very close to the soil. So when the animals will graze, there are less chances of them ingesting these larvae. If they are stall fed, use feeders and waters some on a little bit of height, not very high, but a little bit high from the ground. Avoid overstocking. Too many animals in a very small area will result in easy spread of the disease. And if a new animals are introduced into the flock, then you should rest them for 30 days and aggressively they should be dewormed. They should be dewormed aggressively. And the best is actually you should go for zero grazing. If you can go for zero grazing, that is the best. No grazing, very less chances of ingestion of larvae. So if you are adopting this zero grazing, then it's easier to control the disease. Then important thing is management of the animal sheds. Even if you are doing zero grazing, then it is recommended that the floor of the shed should be raised. You can see that the floor on the first floor is actually perforated. There are small holes into it, and all the droppings they will drop down and get collected on the ground floor. So there will be no contact with the feces in these goats, and even if one or two of these will be harboring the infection, there are very less chances of spread. So that is very important aspect in this case. You should follow zero grazing and zero grazing housing of the animals all the times, and that too in on the raised floors. That is very important. Then another important aspect is if you cannot practice zero grazing, you have to allow your animals to graze. Then the second is pasture management. And frankly speaking, now we do not have any pastures. There are no pasture, but still if the animals are going for grazing on some open areas, you consider them as pastures, and you should use clean and safe pastures. What what are these clean or safe pastures? Which are not contaminated with the larvae of the parasites that affect sheep and goat. And what are these clean pastures? Pasture will you call as a clean pasture that has not been grazed by sheep or goat for the last 6 to 12 months. So pasture resting for at least 6 months will eliminate the larvae from there. If at all they were there, they will be they will die during this time within 6 months. So if a pasture is given rest for 6 months, that is called as clean pasture. But can you have you that much land that you rest a land for 6 to 12 months or a pasture grazing area for 6 to 12 months? Practically not. So what alternative you can do? How to produce a clean pasture? You can graze horses and cattle on that pasture. And then after 6 to 12 months, you again bring sheep and goat into that pasture. The gastrointestinal nematodes of horses or cattle, they are different from those of sheep and goat, most of them. So they will not be transmitted to these animals. Or a clean pasture is one from which is hay or the crop that you have put has been removed and the pasture is rested for some time. Or the clean pasture is one which have been recently established or renovated by tilling it. Frequently till the pastures, then the larvae that are into the soil, they will be exposed to the sunlight and they will die, and the pasture is again fresh. You can again sow some vegetations there and allow your animals to graze. Then Rotational grazing sometimes help to control parasites, but rotational grazing can sometimes also be harmful. It is not always beneficial. So rotational grazing, it depends. And lastly, I told you the sheep, the important thing is when you cut the forages, how much to cut. You should not be allowed to graze on the forages which are shorter than 2 inches long. So it is generally said that these larvae can migrate on the glass blades up to two inches. So if you have some forage which is long, 8, 9 inches, 10 inches, 12 inches, a feet like that, then the animals will graze on the upper inches, not that it will not go up to the lower ground. And even if you cut the pastures, leave this lower 2 inches because maximum larvae will be found in this part. So this is another important thing. Then after this, two important aspects: one, the management, the housing management; second, the grazing management; the third and the most commonly used is the use of anthelmintics. Four broad classes of anthelmintics they are used in India. One, benzimidazoles; second, imidazothiazoles; third, salicylamides; and fourth, avermectin. Benzimidazoles, the most commonly used are the fenbendazole, followed by the albendazole. All everyone knows about these drugs. Albendazole, fenbendazole, they are to use for controlling GI nematodes. But because of their overuse, problems are coming. People shifted from these benzimidazoles to imidazothiazoles like levamisole. Again, problem of resistance has come up in these. Then the people shifted to the recent drugs, that is the ivermectin, which are now being used very frequently. And the problem of anthelmintic resistance is coming with these ivermectins also. And lastly, the closantel, which is not effective against all the GI nematodes. It is effective against the GI nematodes that suck blood, like Haemonchus contortus. So it will be effective only against Haemonchus. Besides Haemonchus, it will also be effective against parasites which are sucking blood, like fistula. So this is another drug that can be tried in for control of Haemonchus infection. But I think you all know the doses of these drugs. The dose is very important. Fenbendazole in sheep and goat, sheep particularly should be used at the rate of 5 milligram per kilogram body weight, not below that. Albendazole 5 to 7.5 milligram per kilogram body weight, not below that. Levamisole again 7.5 milligram per kilogram body weight. Ivermectin 200 microgram per kilogram body weight. These are the doses. All of them can be used by oral route. However, ivermectin it is available in the injectable forms. Oral form of ivermectins are also available. If used as a oral form, the dose of ivermectin should be raised to 400 microgram per kilogram body weight. It should be doubled for using. That is important. The dose is very important. If you are underdosing your animals, you are inviting problem. You are making the parasite resistant to the drug. If you are overusing these drugs very too frequently, you are using these drugs, you are again inviting problem. You are inviting the problem of anthelmintic resistance. So what are the important things that you should consider while going for anthelmintic resistance for anthelmintic treatment? One, avoid underdosing. So how to avoid underdosing? Sheep and goats, they generally are not treated one animal. There are flocks of these animals. If you are treating a single animal in a household, there are 4 animals, then it is better to get an estimate of their weight and then treat them. If it is a flock of sheep, there are 500 sheep in a flock. It is not possible to weigh the animals individually. So what we do is we generally divide that flock into 3 or 4 parts. Suppose 400 animals, 100 animals each. Then in each group, we identify the animal which is the largest one. जो सबसे बड़ा animal है उसको identify करते हैं and then we weigh. And all the animals in that group, they are dosed with the dose rate that is calculated for that weighed animal, the largest animal, I will say. So it will definitely avoid underdosing. Because we have weighed the largest animal, the heaviest animal in that group, and we are dosing all the animals as per its dose. The total dose for the heaviest animal comes to 1 gram, then dose all the animals in that group with 1 gram. A little bit of higher dose is not going to cause any problem in these lower weight animals, because these drugs have a very wide margin of safety as well. Then oral drenching is always recommended, and it should be delivered over the tongue. Drug should be delivered over the tongue. That is important. Third important thing is fasting of the animal. If possible, you should fast the animals for 24 hours, because it will improve the efficacy of the dewormer. If there is no obstruction or no ingesta or digest in the intestines, the drug will be better absorbed. And do not rotate the classes of drugs very frequently. Do not rotate it very frequently. Make a plan, either rotate every year, or there is a concept, another concept, that use an anthelmintic group of drug which is effective at a time. If in today's time benzimidazole drug is effective, fenbendazole is effective at your farm, use it till it becomes resistant. Don't change it till it becomes resistant. One resistance has established against fenbendazole, then change it. Or the second approach can be, you can rotate it on the annual basis. 1 year benzimidazole, next year from another group, third year from another group, then back to benzimidazole. This will dilute the populations of resistant forms and will help in the slow rate of establishment of anthelmintic resistance. Now, anthelmintic resistance, it is the heritable ability of a parasite to tolerate the recommended dose of an anthelmintic drug. If the recommended drug is 5 milligram per kilogram body weight and the worms, they are not dying, even if below 5% worms are not there, which are not dying, then we say that the drug is resistant. And mind you, it is a heritable property. The worm which has become resistant to a parasitic anti-parasitic drug will transfer this trait to its offsprings also. And another thing, once anthelmintic resistance develops, it is difficult to reverse it back. So it is said that the failure to reduce egg counts of the nematodes below 95% is said to be resistance. Agar 100 में से 95 egg से कम destroy, we say that resistance has established in that form, and you immediately change the drug. And selection and reproduction result in the increase in the frequency of resistant alleles, and the problem is most severe in Haemonchus contortus infecting sheep and goats. This widespread phenomena, there are different types of resistance. There is side resistance, means a parasite becomes resistant to fenbendazole as well as albendazole. So this is within the drugs having similar mode of action, the parasite is become resistant. Then there is cross resistance. A parasite has become resistant to fenbendazole as well as to levanazole. So these are two different classes of anthelmintics, and the parasite has become resistant to both. Then multiple resistance, parasite becomes resistant to different classes. It may become resistant to benzimidazole along with amizole and even ivermectin. Then we say if multiple drugs becomes resistant, the condition is said to be multiple resistance. Now, what are the factors for drug resistance? So I have listed here few factors like ineffective drug use, underdosing, too many mass treatments, biotic potential, the worm, drenching frequency, and too intensive use of the drug. And one more important thing that I have kept is depletion of refugia, that we will discuss in the coming slides. Very important. Now, regarding anthelmintic resistance in India, it is a widespread phenomenon nowadays, particularly in the intensively managed sheep and goat. There are hardly, very few farms are left that are where this problem is not there. However, in the semi-intensively or extensively managed sheep and goat, the problem is still under control. It is not that rampant. But in the organized sector, in the intensively managed farms, it is a big problem. It has been reported from all the states, widespread in North India, all the states, South India, almost all the states, Central India, everywhere in all the parts of India, the disease has been, this problem has been, and it has been reported against all classes of anthelmintic salts. It has been reported against benzimidazoles. It has been reported against levamisole group. It has been reported against ivermectin group. These are the three major drugs that are available, group drugs. Then how to test this anthelmintic resistance? It is itself a very big topic. Anthelmintic resistance. There are different types of tests. In vivo testing can be done, or in laboratory testing, that is in vitro testing, can be done. In the field, you can go for in vivo test, which is known as FECRT, fecal egg count reduction test. What we do is that if you are suspecting resistance, you collect the fecal samples from that animal and conduct the EPG. You estimate the eggs per gram of the feces. Once you estimate the egg grams per gram of feces, you then treat the animal with anthelmintic. So that reading that you get on is the 0-day reading. Before treatment, you take the reading. Then after 10 to 14 days, you again collect the fecal sample from those animals and estimate the fecal egg counts. If the number of eggs is reduced by more than 95%, then we say that the drug is effective. If the egg number is reduced less than 95%, then we say that the drug is has become resistant. There are certain in vitro tests that can be conducted in the labs, and these includes egg hatch assay and the larval development assay. Beside these, some molecular tests like allele-specific PCR assays are now available for use in the laboratories, and they require skilled manpower. All of these tests, they require skilled manpower and can be used in the laboratories. Now, once established, it cannot be reverted back. Then what to do? How to prevent it? My animals, they have become resistant to the drug. Should I stop rearing the animals? No. Why should I? So what will I do? So you should always follow epidemiology-based preventive programs. You should know the epidemiology of the parasite in your area. What are the times at which these parasitic infections at a higher rate? What are the months during which this parasite is more commonly transmitted? Which are the hosts which are more commonly transmitted? So if you know all these things, you can devise a parasite control strategy. Yes, you can say parasite control strategy can be devised. So the objectives of these treatment and management programs should be to remove the worm burden before they reach pathogenic level, prevent excessive intake of larvae from the pasture, prevent excessive contamination of the pastures with the eggs, and you should have a knowledge of manage the cases, the times when there is periparturient rise in the egg counts. Now, in case of Haemonchus contortus, what is this periparturient? When around parturition period, just before parturition or after parturition, the egg counts immediately increase. So we should avoid grazing of the animals during these periods. These animals which are showing this periparturient rise phenomena, we should avoid grazing of such animals on the pasture land. Then always practice refugia. Now, what is this word refugia means? Now, in general, I will say refugia, it is an area in which population of organism can survive during a period of unfavorable conditions. But in context of anthelmintic resistance, a refugia is referred to an untreated host and environment that allows the maintenance of drug-sensitive parasite in face of drug exposure. So what does this mean? We should identify the animals in a flock which are not showing clinical signs of the disease or which are having very low counts of the parasites in their body based on the examination of fecal sample. And we should leave these animals as such. We should not treat these animals. Beside these animals, all the animals are treated. So we should identify a few 10, 20 animals in a flock which are quite healthy, which are not showing any clinical signs. They are passing eggs, but the egg counts are not very high. So we should identify such, they are not showing any clinical signs. So we should identify these animals and do not treat them. leave them in the flock like this. Because once you are treating other animals, they will be exposed to anthelmintic and the resistant parasites will survive. They will pass their eggs in the droppings. And if somehow these animals which are left from deworming, they are also passing eggs. So they are susceptible eggs. The eggs that are passed out from a treated animal, they are resistant eggs. So there is dilution of the resistance. So this is an important way to practice refugia. It is very important. And that is actually what is required. Then the third thing is targeted treatment and targeted selected treatment. What is targeted treatment? Some flocks are left untreated while other flocks are treated to maintain a selected susceptible population. So this is what I am telling you how to maintain refugia. Refugia के लिए कैसे animal है? How to maintain refugia? Either you go for selected treatment or you go for targeted. In selected treatment, target selected treatment, treatment is given to individually a determined animal, which is based on GI monitoring system like FAMACHA chart. FAMACHA chart is FAFA Malan chart. This is a specialized chart developed for monitoring anemia in the animals. And based on the anemia, level of anemia, you decide whether to treat the animal with anthelmintic or not. I will show you in the coming slide. Only a small portion, that is around 5% of the animals, they are left untreated while the rest are treated. And they will, both these methods, they are directed towards minimizing the impact of anthelmintic resistance. The chart that is showing, that you are seeing on the right side is the Fafa Malan chart. And you can see the varying degrees of anemia, right from the number 1, where you can see the conjunctiva is reddish in color, second pinkish in color. You leave these animals. You don't treat these animals because their blood levels, hemoglobin levels are quite good. So the parasites in these are not actually causing harm to the host. So you can leave them for treatment. The third, you have to decide, which is very light pink in color. Very light pink. You decide yourself whether you want to treat or not. While the fourth and the fifth, where there is severe anemia, the conjunctiva is almost whitish, you have to treat. Use this chart to determine which animal is to be treated. Then comes a 5-point check formula, I will say, for target selected treatment. So you look for 5 things. Now things are now changing, and now they have said that instead of looking only for anemia, you also look for these 5 points. The eye, first, I already told you, FAMACHA chart. This gives an idea of blood-sucking parasites like Haemonchus and liver flukes, fasciola. You look for the back score, the back of the animal, the body condition score. And this is applicable for all the animals. Then comes the rear. You look for the rear, the DAG score, the fecal soiling, evidence of scoring. These things you need to check, and all these gastrointestinal nematodes will have these kinds of symptoms. The fourth, you look for the bottle jaw condition. And lastly, you also look for the nasal discharges. Particularly, this is not important for gastrointestinal nematodes, but it is important for the parasites that inhabit the nasal passage, like nasal port. So if you follow this 5-point check based on what kind of condition is in these parts, you can decide which animals you want to treat with what anthelmintics and how. This is important. The 5-point check is an important method for target selected treatment. Then alternative methods of control are also there. But they are not, right now, they are not much used in the field conditions. They are under developmental stages. Like in biological, there is a method for biological control of the parasites. And for gastrointestinal nematodes, it is the nematophagous fungi. There are certain fungi which actually trap the larvae of Haemonchus and do not allow them to spread on the pastures. So these nematofungi, they are fed to the animal. The spores of these fungi, they are fed to the animal. They are non-pathogenic actually. nematophagous, non-pathogenic fungi. They are fed to the animals, and the spores will be passed out in the feces of the animal. The feces will also have eggs. So when they come into the environment, the fungal spores from the fungal spores, hyphae will start emerging. And on the other hand, the larvae from the eggs, the larvae will be produced within the fecal pad. Both things are occurring in the fecal pad, and these hyphae will then trap the larvae of these gastrointestinal nematode and prevent their spread onto the pastures. So this is how these nematophagous fungi work, and they are important biological control agents, though not they are, I think most none is available commercially right now. Another approach can be you can breed certain species, or you can breeds of those animals which have shown some genetic resistance to these parasites. There are some breeds of sheep and goats have been identified. Particularly in Africa, they have East African, they are red Masai sheep. In India, we have Jamnapari and Garol breeds of goats particularly, which they say they are quite, I will not say they are genetically resistant, but they are said to be resilient to the infection. Resistance and resilience are two different terms. Genetic resistance is where the parasite is unable to develop, but resilience is the host in which a parasite develops, but it cannot produce a clinical disease. So these Jamnapari and Garol breeds of sheep, they are said to be resilient to the Haemonchus contortus. Last is the immunoprophylaxis. Control of parasitic infections by use of vaccines. And for long-term parasite control, this seems to be one of the best methods. But unfortunately for parasitic infections, none of the vaccines are commercially. For GI nematodes, there is only one vaccine that is commercially available, that is Barbervax, and that vaccine is not available in India. And this vaccine is specifically for Haemonchus contortus. So, but this is not available in India. The take-home message, what I will say is that it is important to control gastrointestinal nematodes. We should go for grazing management, and the best is go for zero-grazing. And besides grazing management, you should integrate the grazing management along with the other measures of parasite control, like chemotherapy or chemoprophylaxis, and non-chemical methods like nutritional management or housing management and other things. So all these things needs to be incorporated into one program so that you can go for a effective gastrointestinal nematode control.